Okay. Today we're going to continue the discussion of the human genome, or genomes, and we're going to talk about your personal genome, and we're going to talk about human origins, and hopefully we'll see how it goes. Maybe I'll even have time to talk about race, which is something which I enjoy very much talking about in the context of DNA. Okay. I gave you another lecture. It's called Lecture 5.1 because I upgraded Lecture 5. And I wanted you to have, so follow along with me with respect to that. Okay, we know that there's two genomes in human cells, these marvelous things, one in the mitochondria and one in the nucleus. And I talked last Thursday about this incredible organelle, the mitochondria, which makes all the energy and has its own genome. And I discussed the fact that when mutations occur during DNA replication of that semi-autonomous genome, it can lead to quite serious diseases. And I pointed out, Anna and Derek, that the inheritance of a gene which causes a human disease in the mitochondrial genome is very unique because it's passed on from mothers to all the children. And that even though a father may have the disease that he got from his mother's mitochondria, he won't pass it on to his children because of the fact that most of the mitochondria come from the egg. And so there's a very unique pattern of inheritance. And so I went through those different things. And then I think I wound up talking about, in fact, I know I wound up talking about the human genome. And I discussed the fact that the human genome has been sequenced now for almost a decade. And I talked about how much it costs. And I talked a little bit about how much it would cost today to do this. And we're going to pick up on this topic a little bit later on. And I talked a little bit about how you can't do any of this DNA sequencing without genetic engineering. And I emphasized the point, and I think I did, Thomas, and remind me if I'm wrong, that genetic engineering underpins all of this technology. And ultimately, in order to sequence anything, you need to have a series of clones. And I think I also discussed the fact that when you have a series of clones in the context of factor 8 that represent overlapping regions of each chromosome, you can sequence each chromosome, and then you can map them, and you can have the sequence of the human genome. And then I talked about the fact that all of these closely related creatures, including a tree shrew, have been sequenced. And I said that we can learn an awful lot about ourselves, what makes a man rather than a mouse, or a mouse rather than an elephant, from comparison of our genomes. And I pointed out the fact that our genomes are very closely related. And I said if this doesn't say that towards the end, going into more details right there on the human genome. So let's pick this up and set the stage. The human genome has 3.2 times, can you hear me, John?
Hear you fine, Bob.
Great. It's a very, very large genome. But what's so ironic about this genome is that only 1.5%, think about that, 1.5% of this gigantic genome specifies proteins. And as far as we can tell, plus or minus a few thousand, Eden, there's only about 25,000 genes in your human genome. But we know that there can be a lot more proteins in any given cell because we talked about the fact that there are these introns and exons, and you can have alternative splicing before the messenger RNA is made so that any one of these genes can make 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different messenger RNAs and 10 different proteins. And so you can have much more than 25,000 proteins in any given human cell. And in fact, we do. So that's an important fact. But what's astonishing is that this genome, as I said, is so large, but only such a small percentage of it specifies proteins. And what is the rest? Well, the rest is the introns, that is the non-coding regions inside the genes. And I'm going to go over this a few times. The rest, Max, is the regions between genes because genes aren't totally next to each other in a very compact form in the human genome. And the rest are these magical things called repeated, like repetitive DNA sequences. And what you can see here is that repetitive DNA sequences can represent almost half of the DNA in the human genome. So almost half of the DNA in the human genome represents a sequence that is repeated, meaning many, many times that exact same sequence is present in the human genome. And this is a characteristic feature of all higher eukaryotic genomes. It's not a feature of prokaryotic genomes, but it's a feature of eukaryotic genomes. And it's really, in some respects, considered to be an evolutionary relic. It's considered to be a relic of DNA getting into, let's say, the human genome and just perpetuating itself because it's self-replicating in many respects. That is, it's replicated along with the rest of the DNA. But there's two kinds of these repetitive sequences. And again, I'm going to go over this again conceptually, as you can see. One kind are these things called transposable elements. These are pieces of DNA in the genome, like hopscotch that can hop. Literally. Over evolutionary time, they move. Not over your time, not over in our lifetime, but over evolutionary time they move because many of them have a gene in them. That gene specifies an enzyme, and that enzyme has the ability to excise this piece of DNA from the chromosome, allow it to move, and pop itself down in another part of the human genome. And they're called transposable elements. What do you think, Mike, about the consequences of these transposable elements over evolutionary time? What would be your hypothesis?
I would hypothesize that you could trace the changes of them and therefore know how certain organisms and species have
Let's
evolved.
just stick to the human genome.
The human genome, you
Yeah.
could see relationships between groups of people based on
Transposable elements, you think?
where they've moved over.
So
Long
one, you
periods of
can trace them. They do. You can trace them. But what do you think would be a major consequence of these things moving around your ancestral's genome millions of years ago?
It would interrupt it.
And what happened? What are the consequences of that?
You'd have proteins that were no longer being created correctly.
Can you say that aloud?
You'd have proteins that were no longer being
So
created
it's
correctly.
possible that these things could move around the genome and land in a place which they're not supposed to be landing in.
Yeah.
Is that correct?
Yes.
So if they land in a particular gene, that gene might not function anymore. Is that correct?
My hypothesis would say
How
so.
might these things moving around the genome contribute to the evolution, Lauren, of form? Let's go way back in time. How might they contribute to the evolution of forming a new arm or forming another eye? What do you think?
Well, depending on where the transposon landed, it could alter the gene and alter the protein that was made to alter the shape of...
We already said that, though. But what about causing something big, like the formation and the ability of an arm to form? How do you think a transposon might do that?
Could it go into like the regulatory...
Go ahead.
Go into like the switches? Where
Yeah, what if it went into the switches and it happened to turn off, let's say it happened to turn off a certain gene that was responsible for, let's say, eye formation. But instead of eye formation, maybe that transposable element carried something else with it that was a different switch that turned on those genes at a different time in the development of, let's say, these primitive hominoids, and maybe switched on genes at a certain time that may allow it to establish an arm or something like that, or a finger. And so in many respects, the ability of these things to move around the genome may produce evolutionary genetic novelty. In many cases, they're going to produce things that are going to lead to lethality and death, but they can possibly lead to some new genetic utility and some genetic novelty. Now remember, these things are happening over hundreds of millions of years. Yes, Julie, go ahead.
Well, is that why we don't have tails?
That's a whole other story for another time, okay? Yeah, the point here is that these things might have some utility. rather than just moving around and mutating our genome. So the vast majority of these things are in the class of transposable elements. But there's another class which is more, let's say, dear to your hearts in this class, and that is these things called simple sequence repeats. And those are the repeats that you know as VNTRs. And those represent about 3% of the DNA in your genome, and they're very useful for individual identification, for all the things that we've been talking about, simply because of how they're organized in the genome. And I'm going to come back to this in a second. The interesting thing about sequencing the human genome, as I said on Thursday, is that half of the genes in the genome, we don't even know what they do. And I think I said to you that that means that Daisy and Aicha and Kelly, and I said this, are going to have a lot of things to do for the rest of their scientific careers, because that means that we don't know the functions of 12,500 of these genes, and that means that someone could spend a lifetime just working on one gene. And so there's a lot of things that we need to learn about the human genome, just simply in terms of what these genes do and what things they specify inside your cells and inside your body. But the other half, we know what they do. You know, regulatory proteins, energy, membranes, all the structure of the cells, all these different kinds of things. We have a good idea of what they do. And how do we know what they do? We know what they do because of what you learned about the second week of class and this thing called bioinformatics. And remember, once they sequence the human genome, they take those sequences and they put them in that BLAST server, and they say, are there any other DNA sequences anywhere on the face of this earth that people have sequenced and know what the protein is that it codes for, and do they match anything in the human genome? And by doing that, they start finding what these random DNA sequences are. Not so random, but they're just simply DNA A's, G's, C's, and T's. And by doing that, they can start finding out what proteins these genes encode. But there's another way in which they can do this. And this is very important to consider conceptually, Chris. If you have an unknown human gene, how do you find the function of a gene you don't know what it is? Remember purple and green cabbage? What were purple and green? What was the relationship between purple and green cabbage? I keep coming back to it.
The purple cabbage mutated so that,
yeah,
like,
formed a green.
yeah.
So the relationship between purple and green cabbage was mutation. Mm-hmm. How do you find the function of an unknown gene?
You could.
Wow.
Yeah.
Yeah. Hand it over to Dora. She's smiling here, dreading that I call on her, and now I've gotten an opening. How do you find, given purple and green cabbage, there's a mutational relationship between the two, antibiotic resistance, antibody sensitivity, antibiotic sensitivity. How about round and smooth, virulent and avirulent? We've talked about all kinds of alternatives. How do you find the function of an unknown gene? What's the only way in which you can do it, other than a search on a gene bank site?
I would make a hypothesis.
Go ahead.
And test for it.
Well, what's your hypothesis? How do you find out the function of an unknown gene? You're turning red.
I don't know.
Well, you just said it, Chris. Can you help her out? What's the word that we're talking
sequence?
about? Starts with an M. Ben?
Oh, I have one, but
What's your hypothesis?
I think you can use a knock-in procedure.
Well, we don't know what that is yet, but what is the conceptual word that you have to look for?
Mutation?
Yeah, you need to have a mutant. You can't find the function of a gene unless you disrupt it in some way and see what function it disrupts. And you think you can do that, Stephen, in humans? No, it'd be pretty unethical to start knocking out human genes. And so what's the most closely related creature you can do it with, Thomas? I think you'd have the animal rights people coming after you pretty darn quick if you did that. What do you think? What do we talk about, all these human and mammalian genomes? Amelia, do you want to take a stab at that with the microphone? Yeah, do you? What do you think the creature is that you would work with to find out?
Bacteria, like E. coli.
It's not very related to humans. What would be most closely related organism to humans you could start working with?
Rat.
A what?
Rats.
You're close. It's a mouse. Mouse and humans only have about a 1% difference in their genomes. And I just showed you in one of these slides, which I'll go back to, that you can line up. There it is. There's mouse chromosome 12 and human chromosome 14. You can line up all these genes. And Daisy, if you don't fix this, it's going to drive me, or Aicha, it's going to drive me nuts. I think it needs new batteries. So I'll use this red one. In any case, look at the lineup of those particular genes. So one can theoretically then use a mouse, identify a human gene from the sequencing, find by using a blast search the comparable gene in the mouse genome because it's been sequenced, and then knock it out with procedures that we're not going to go into here. In other words, you knock it out by genetic engineering. You add a gene and you direct it to the place in the chromosomes where you want it to go, and it destroys that gene. You know it's destroyed, and then you look and see what the mouse does. And this is a very important model organism for studying human genes because it's the most closely related mammal in which you can start manipulating genetically and by genetic engineering in a laboratory. And it's very good for making cancer models, diabetes models, all kinds of disease models, and finding the functions of human genes. And so we have all of these unknown genes here in the human genome, but you can find some of their functions by working in a mouse. That's not to say that the gene that gives you a particular function in the mouse will give you the same function in a human, but you can do some kinds of things to try and get some correlative types of inferences. Derek, do you agree with that? Just what I said. You were drifting, right? Just a little bit. I know everything. Okay, let's talk about, we have 25,000 genes in the human genome. God, I love doing this. There's more fun in this classroom than I would rather be here than almost do anything. It's so much fun. 25,000 genes in the human genome, look up on that slide, but of the number of genes that we know correlate with human diseases, and of course not all genes going to cause a mutation and genes going to cause a disease, of course. Some of them are going to be lethal. You'll never get an organism. They're going to cause all kinds of really serious defects. But look and see, there's only about 3,000 that we know correlate with Mendelian defect. And when I say a Mendelian defect, I mean something like sickle cell anemia. I mean something like Duchenne's muscular dystrophy. I mean something like cystic fibrosis. I mean something like factor VIII pedigree, of which then you can identify behind that pedigree the gene in the genome that's responsible for that particular phenotype. And of course, for all of those 2,700 to 3,000 genes, you have then markers. What they are, then you can use markers to follow those genes in a pedigree. Kathy, do you understand that? You don't understand that? Right behind you, Kathy on. Okay, how about you, Zhao? Good. Okay, Stephen?
How do we know that number 2,700 if we don't know what half of the genome does?
Yeah, so you know that number of 2,700 because those are the number of diseases that have been identified over the last 100 years by just looking at the outward phenotype of the defect, like sickle cell anemia. I mean, we knew that there was sickle cell anemia before we knew what the cause of it was. And then by all of these studies that have been carried out, not necessarily at the DNA level, but at the biochemical level, at the cellular level, at the protein level throughout all of these decades, and then at the sequence level, you converge on all of those things, and you tie all those pieces together, and you say the sickle cell anemia gene is caused by a defect in a globin gene, which is a protein in the blood. And so it's a lot of different studies that come together to make these correlatives. But clearly, there's many genes that cause diseases. There's about 1,000 of them, I believe, so far, that we have phenotypes for, but we don't even know what the gene is. And we don't know what the gene does Because remember from your discussion last week, you can have markers for genes, but that doesn't necessarily mean they're in the gene. That doesn't necessarily mean you know what the gene is. And so many genes that cause human diseases haven't been identified. Even though the sequence is in the genome, we haven't made a one-to-one correlation between that disease and the gene. Is that clear? Okay. So we only know a few thousand of them. So we have a lot of work to do. And here are the ones that we know, and you've heard of many of them. In any case, here are the ones that we know. Here's what we've talked about. Hemophilia, we've talked about sickle cell anemia, we talked about cystic fibrosis, and all the others. And when I say here are the ones we know what the disease is. We can recognize it in a family. We know what the gene is. We know what the protein is. And we know when you disrupt that gene, you cause a very serious and deleterious disease. Many of them, we don't know what they do. Now, let's go back and look at this. Chromosome, right, Faria? How many double helical molecules are there in a chromosome, Faria, that hasn't been replicated? 1, 2, 5, 10. What do you think? Where's the microphone?
2?
In a chromosome that hasn't been replicated, how many double helices are there? Is that a chromosome that's been replicated or not replicated? Rose. Has that chromosome, is that a picture of a chromosome before DNA replication or after DNA replication?
Because it's one-sided, I'm going to say
Well,
after.
I'm not asking, what is it, before or after?
After.
It really is. What is your evidence for it being after DNA replication?
Okay.
Did you guys have a discussion? Did you have a discussion last week? Are you sure? Did you have a handout? I remember this on the handout. Is it before or is it after? Taylor?
Before.
Yeah, it's before. Remember, replication means you replicate the genome. So each chromosome has one DNA molecule until replication. And the DNA replicates, and we have 2 parts. Wake up, Mike. We have 2 parts to the chromosome, and it goes 1 to each cell. And so when you see a chromosome with 2 chromatids, when you see a chromosome that looks like this, as opposed to just this, that's after DNA replication and that's before. You have to think about how things are inherited from cell generation to cell generation. So this is a chromosome that hasn't replicated yet. This is a chromosome with one continuous DNA molecule from one end to the other, and we've discussed that. It's a DNA molecule which is wound up into a little ball. And why is it wound up into a little ball? It's because if you take the DNA in the human genome and you lay it end to end, it's about 1.2 meters in length. But the size of a nucleus is only 1 times 10 to the minus 6th meters in diameter. And so that means that these chromosomes have to wind the DNA like yarn on a little spool in order to get it inside this really tiny nucleus. And furthermore, the chromosomes, in order to move from cell to cell during division, if they weren't compact, there'd be a greater chance of these chromosomes breaking. And so they wind in order to facilitate the division process much more readily. And so we have a chromosome here that's pretty much wound. And when it's wound the shortest is at this phase of cell division that I know you've had in biology somewhere way back when called metaphase. Remember prophase, anaphase, metaphase, interphase, it's all mitosis. You remember that sometime way back when. I know that everyone has this, even in 6th grade at some point. What?
I could draw you a map.
Yeah, I know. Okay, but try and make these correlatives. So here we have the DNA molecule. Now we're talking about the sequences in this molecule. And here is our garden variety gene. Here's the 5' end and the 3' end. And remember, sense and antisense, transcribe, template, all of those things, activators, promoters, All of that stuff would be up here, upstream, downstream. But remember, the DNA is all connected, right, Thomas? It's all connected. So here's going to be a distance between this, and here's another gene right over here. And here's another gene right over here. And here's something here, what I'll come back to. Here's something here, and here's something there. That gray little rectangle is exactly the same sequence, Marvin. But it's something that's now repetitive. Notice that this little gray rectangle, sequence 1, sequence 1, sequence 1, it's an identical sequence, and that's why it's called repeated. The genes are not repeated. You'd never get a mutation in a gene if it was repeated. Do you agree with that, Thomas? If I had 10 sickle cell anemia genes and I mutated 1 and they all worked, I'd never get sickle cell anemia, correct? So most of your genes are present only once in the genome, but these repetitive sequences are present in many different places. These are the transposable elements. And why are they transposable elements? Because they're present here, they're present in a different place here, they're present in a different place here. They may be on different chromosomes, but it's the exact same sequence, just replicated and repeated many hundreds, and in some cases, millions of times in the human genome. On the other hand, you keep going down the chromosome, and you find these things right here. Those are also repeated sequences, a different sequence. This rectangle is sequence A, repeated 3 times on that chromosome. This is sequence B, repeated 1, 2, 3, 4, 5, 6, 7, 8 times on that chromosome. And those are your VNTRs, variable number tandem repeats, called variable number because they can be different in different individuals, and we're going to talk a lot about these today. And in the case of this DIS80 locus, the one we use for your class DNA fingerprint, the repeat is 16 nucleotides. So the core repeat is 16, and then it can be repeated any number of times depending upon the individual. Here's another figure that shows the same thing. It shows that your coding sequences are only 1.5% of the whole genome. It shows that the transposons are 45%, and it shows that these little repeats, these simple sequence repeats, these VNTRs, are 3% of the genome. It's a lot. And there's various, and here's the key here, there's transposons that are different in sequence. Do you have that, Eden? There's transposon A, transposon B, transposon C, totally different sequences. There's VNTR A, VNTR B, VNTR C, totally different sequences. So if you want to detect VNTR A, you need A probe. If you want to detect VNTR B, you need a probe for B, et cetera, et cetera. So these are different sequences, but they're sequences that are just repeated either randomly in the genome or next to each other in a particular way. Now, from the point of view of DNA fingerprinting in the law, from the point of view of DNA fingerprinting in paternity, from the point of view of DNA fingerprinting in classification of plants, that is, individual plants, from the point of view of DNA fingerprinting in what we'll be discussing, these simple sequence repeats are what's important. That these are VNTRs, but they're simple. AC, 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 80,000 of these things somewhere in the human genome. They could be repeated 5 times, 10 times, 16 times in any one location. But in another location, they could be repeated any X numbers of times. So it's these different simple sequence repeats which are important. You have a question.
Yeah. What's the difference?
That was inherent in your hand. Go ahead.
What's the difference between a simple sequence repeat and a transposon?
What's the difference between a simple sequence repeat and a transposon? Let's see. Brian, do you want to answer that question for Marvin? What's the difference between a simple sequence repeat and a transposon?
A transposon is a piece that can move, and then a simple sequence repeat is just a VNTR, a very simple one.
So the transposon actually has, in some cases, coding information in it. So the transposon could be a sequence that might code for an enzyme that could cut it out of that location of the genome and plop it into another location. Most of the transposons in the human genome are defective. They don't move. They've been mutated so that these enzymes that cut them out and move them together don't work anymore. Okay? So you don't have to worry about your genome blowing apart Marvin. But the VNTRs don't code for anything. In fact, many of them are very simple. AC, 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 ACT, 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 ACT. They have no coding function, and they never move. And they're next to each other. Transposons, because they move, can be scattered throughout the genome. They're dispersed. OK, what about this variability? So let's try this. Well, I already did that. So let's go for this. Different forms of the gene are called alleles. Yes or no? One, two, three, four, five, stop. Great. Who said no? Who's going to be brave enough to say there was no? Hiromi, you said no? Positive? Elaine, did you say no? Lauren said no. Taylor said no. Someone's just pulling my leg. I think that's what that's all about. In any case, the point here is that you're absolutely correct. Here they are. And here we're going to review this again. Remember the tomatoes that I showed the first day of class. Remember, Marvin, when you were so nervous the first day of class, which you're not anymore. Here we have a chromosome. Before or after DNA replication, Eden? How come there's two of them? I can't hear you. John can't hear you.
There's two of them because there's two. We have.
Where do
two
those
sets
two
of
come from?
chromosomes. Your mother and your father.
Okay, so blue from mom, green from dad, correct? And DNA replication hasn't taken place. This little thing here called gene A, is that one position of that chromosome, Eden? Speak into the microphone.
Yeah, a look.
Does it have a definitive, specific DNA sequence?
Yes.
And it's different than the sequence down here, correct?
Yes.
Does this chromosome have polarity?
Yes.
5' to 3', let's say. Does it have one DNA double helix or 7?
One.
Exactly. These two chromosomes that come from the mother and the father, could they be identical in DNA sequence? Could they be?
They could.
Absolutely. Are they identical in DNA sequence?
In DNA sequence, probably not.
No, they're not, because notice here we have this locus, and that locus can mutate. And how many different forms of the same gene can we have in any given individual, Eden? You're doing great.
2.
You're sure?
Well,
you're sure? How many
3.
forms? 3?
Well, there's
3?
Sorry, how many different forms do you
of the
mean?
same gene? How many different forms of a same gene do you have in you, potentially? Maximum number.
Of a gene, maximum is 2.
Exactly. How many in this room?
Maximum 2.
In the whole room?
Yeah.
Forms of the same gene in this?
Of the same gene, yeah.
Just only 2 forms.
Well,
how come we look like 30 or 32 different people?
Well, of one gene or of...
One gene, this is why I'm going this, these are alleles, right? Michelle, how many different forms of the same gene could we have in this room?
2 times how many people are in here?
Yeah, we could have as many as the people in here. That's exactly correct. That's why we look differently. And what are those different forms called, Michelle? What are the different forms of the gene called? What's the word for it?
Mutation?
No, there's a specific word. Different forms of the same gene. Alleles. I've been talking about this since the first day of class.
Alleles.
Yeah, exactly. They're alleles. And so you only have 2 different forms of the same gene or 2 alleles inside you, but in a population you can have thousands, millions, potentially millions of different forms of the same gene. But now what I want you to think about, Chris, more than anything, is that alleles have nothing to do with genes. We only call them alleles because you see brown eye and blue eye and blonde hair and black hair and alternate phenotypes. But any difference at any locus, meaning any position of the genome, is an allele. So you can have an allelic form of any DNA sequence in that same position. Is that clear, Julie? That's critical to understand. So that any position along this chromosome, if you have a DNA sequence difference, even though it might be not be in a gene, that would be an allele. That could be an allele of each other. And you can use that as an identifier for that individual or that population or that ancestry. And in fact, most of the differences that we measure between individuals or populations have nothing to do with genes. The DSI-80 that we genotyped you with has nothing to do with a gene. It's not a gene, it's a VNTR. It's a small, simple sequence repeat. And so, but there are differences, and those are alleles. And so anytime you have a difference at a particular chromosomal location, it's a difference. And so you can have an infinite number. So is that clear? So here we have gene A, gene B, gene C. In this case, we have gene C, both exactly the same forms in father and mother chromosome. Here we have different forms. Here we might have the same forms, but you can have identical or differences or as many as you want. The main point here is that these differences between chromosomes, between individuals that arise by mutation, and we'll go over this in a second, most of these differences are in parts of genes or between genes that have nothing to do with anything. And you can notice that here's two cystic fibrosis genes, and these are the differences in base pairs, nucleotide differences, that are distinguishing those two cystic fibrosis alleles. In other words, you can have mutations, and we've talked about this, called single nucleotide polymorphisms, that lead to nucleotide base pair changes between these two, essentially, chromosomes. And so you could be perfectly normal with respect to cystic fibrosis because there's no change in the exons, no change in the coding portion of the gene, but you can have still nucleotide differences in parts of the gene that don't do anything or in surrounding areas that don't do anything. And these are markers for individuality. Is that clear, Mike? That's very important. Stephen, is that clear? And these occur randomly in DNA replication. And these markers are critical for either identifying a gene. So for example, any of these particular polymorphisms could be used to mark the cystic fibrosis gene. Would you agree with that, Thomas? Because they're in the gene. And if you can follow it, it marks it. Or any of these markers could be used to identify your mother's chromosome or your father's chromosome. Agree? Or potentially your grandmother's chromosome or your grandmother's chromosome, and going way back. So they can be used as identifiers of genes and marking disease genes to follow them for genetic counseling and other purposes, but they also can be used to mark individuals. Catherine, do you understand that? It's critical. Taylor, do you understand that? This is really, really important stuff. Does everyone understand that concept? Derek, do you understand that? Okay, so here are the different kinds of polymorphisms that can occur. We have single nucleotide changes. Here's one sequence of, let's say, your mother's chromosome. Here's another sequence, a part of a chromosome of your dad's, and it has one nucleotide difference due to mutation. Single nucleotide change in replication is called single nucleotide polymorphism, or a SNP. And here we have a different kind of change. Here's a simple sequence repeat. In your mom, it's just AT, AT. There's just two repeats. In your dad, it's AT, 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 6 repeats. And so those can change too, but it's a different kind of marker, but it still marks your mom's and dad's chromosomes or a gene or in between genes or whatever. So they can be used as markers. Believe it or not, anywhere in this room, Anna, anywhere in this room, Hiromi, Lauren, there is over several million single nucleotide polymorphism differences between any one of us. That's why we look different. That's why we are different. Some of them land in genes and cause differences in phenotypes, so they're different alleles, different forms of the same gene. But many of them don't land in genes. In fact, most of them, as I said, and they can be used by me to identify any one of you. And I did that with the DNA testing in this class. I can identify any of you from that DNA test. And if I use more than one so-called probe, if I looked at more than one loci, it would be even greater confidence, as we'll get into in a second. So the point is, there's a lot of variability between us. Samantha, my woman, what's up?
Okay, so my question is, you're saying that if we have these
just to answer a question, don't say what I'm saying. Just be direct.
Polymorphisms inside non-coding genes, why do those make differences in humans if those
I didn't
aren't
say
being coded?
that. I said that the polymorphisms in the genes cause
what
the
make us
phenotypic
different.
differences.
But
The
things
ones in between the genes or in the non-coding regions can be used to identify individuals.
the differences, but it
But
doesn't
they
make
don't
it
cause phenotypic changes. Okay? Great. And they can be used to trace. That's what makes us individuals. So these mutations occur when? During transcription, right, Eric? Okay. Right, Tiger? All these mutations occur during transcription, right? Dora? Positive about that. So mutations in DNA occur during transcription, occur during translation, occur during splicing, occur during replication. When does it occur? 1, 2, 3, 4, 5, display. Who said during transcription? What is the main? Albert said during transcription. Okay. What would happen if a mutation occurred during transcription? Can you take the microphone?
Then
Not what would happen? Be precise.
everything would be messed up.
Would it be a mutation? What's the definition of a mutation as we've been talking about it? What happens to a mutation? From your mom to your grandma to your great-grandma?
It gets passed.
Yeah,
on.
replication passes it on from generation to generation. If it happens in transcription, you're never going to see it as a mutation. The gene might be transcribed 5 billion times. What are you going to see? You're going to see nothing. So whoever said transcription better go back and read chapter 2 of your textbook, right, Daisy? Because this thing happens during DNA replication, and I spent a whole lecture on that. And so please review that. So the point here is that these SNPs occur by mistakes in DNA replication, and here they are. And what we know is, again, a mistake in DNA replication can cause this GC to change into an AT. And that's a single nucleotide polymorphism. And these are very rare, but they accumulate over hundreds of thousands and millions of years. And they stay there, or they can mutate to something else. So here's a SNP. We call them single nucleotide polymorphisms due to just simple base pair change. And here has something else. Notice, and I'm going to come back to this, that on a given chromosome, notice that there's a, what's the matter, Daisy? Thank you. That there's a SNP here, a SNP here, and a SNP here. And notice that. And so notice that there's chromosome 1A, 1B, 1C, and 1D. And I'm going to come back to that in a few minutes, because that's also useful for marking chromosomes, that is, changes in nucleotides next to each other and that are inherited as a unit. And this kind of situation, as opposed to this, is a marker of ancestry. Because in general, these three changes that are very close to each other in the genome will always be passed on together as a unit. And so when they occur way back in time, let's say in the time of Adam and Eve, they're going to be passed on to all of Adam and Eve's descendants, provided that they don't change. But the original Adam is going to have this little CGG going way back when. And so the point here is that these single nucleotide polymorphisms, once again, can be used to mark real genes, like sickle cell, and trace them in pedigrees. But because most of them are not in genes, they can be used to trace lineages. And what's the most powerful trace of lineages is when they occur together in little clusters next to each other. So you're not just following one single nucleotide polymorphism, you're following 3 or 4 that are always together in a cluster and never, ever, ever break apart. And that's phenomenal and powerful for saying where you come from. And I'm going to come back to that a little bit later on. So there they are. There's your SNPs. There's your polymorphisms. These are all the different things that can give rise to variability in your genome. But what's most useful are these things called VNTRs, SSRs. I'll come back to that, or SNPs. OK. How are they used? As you'll see, in the case of your personal genome, that means my genome, your genome, Brian, Albert's genome. How are they used? They're used to mark genes. They're used to say Samantha, Samantha, Bob is Bob. In this case, there's a SNP that is a single nucleotide polymorphism that's linked to the eye color gene. And there's different forms of this SNP. meaning someone might have an A, someone might have a C, someone might have a T, someone might have a G. It's linked to the eye color gene, and notice we got brown, we have hazel, we have blue, we have hazel, we have brown, and these are predictors of eye color. And so if one can look at this SNP, at this eye color gene called the P gene, which makes melanin, one can predict with reasonable confidence, and I'm going to come back to this, what might be your eye color or the eye color of your children or whatever. And so these particular markers, these SNPs, can be used to mark genes. And you know all this. This is a review. And furthermore, you find them in certain ways that I'm not going to get into, but once you find them, as I said at least 5 times in the last 10 minutes, they're used in 2 different ways. They're used to mark disease genes and follow them and isolate them as well, but they're also used to trace ancestry and lineage. So let me talk about disease genes, because that's what you're most familiar with. Here we have a disease gene. This is what we talked about on Wednesday, Nancy, in discussion. If you have a disease gene, it's manifested by a particular phenotype, like sickle cell anemia, like Duchenne's muscular dystrophy, like cystic fibrosis. There's an outward manifestation of that phenotype. If you use genetic methods that I'm not going to get into that says where this gene is located on the genome, and if you have a marker that is either in that gene or, Thomas, next to the gene, linked to it, never separated from that gene ever, you can use that marker to trace that disease gene at the DNA level, which is very powerful for genetic counseling in families or before birth or before implantation of an embryo, and we've talked about all of that. So the point here is if you have a SNP or a marker in or surrounding a disease gene, and if it's linked 100% with that disease gene, it can be used as a predictive that that individual may carry that disease gene or have 2 copies of that disease gene or whatever. On the other hand, let me flip. If you're working in agriculture and you're trying to select, let's say, for wrinkled and round seeds, that takes a long time. Because if you want to select for round seeds as opposed to wrinkled seeds, you've got to wait till you get the seeds to look at them. But what if you had a marker of those genes so that as soon as the seed germinated, you can take a little bit of the leaf, you could extract the DNA, and you have a marker for that particular phenotype, you could start doing your selections very much more rapidly. And so in this situation, it's exactly the same as this. We're not dealing with a disease gene, but we're dealing with an alternative in the phenotype of a seed, but we have a marker. Here's the marker. We have a marker for the round seeds. We have a marker for the wrinkled seeds that you can detect using PCR or a DNA blot. And we have a marker for a heterozygote. And by using these molecular markers that correlate with the phenotype, no different than a disease. Do you understand that, Faria? It's exactly the same thing. You can use this in agriculture and breeding. It's called marker-assisted breeding, and it really speeds things up because you don't have to wait until the seeds form. As soon as the generation of the seed germinates, you can look at the DNA and say, this is the plant that we want to select. This is what we don't want. So they're very powerful both in human genetics and in agriculture and in all kinds of things. Okay, you know all of this, so I'm not going to spend a lot of time on it. We have a single nucleotide polymorphism in a restriction site. The polymorphism renders that restriction site inactive, meaning the restriction enzyme can't bind to the site. That means what we have here is one band, one DNA band, because it can't be digested with the restriction enzyme. So that's this allele. The other allele can be digested. We get 2 bands. And if we know that that is linked to a particular disease, like sickle cell anemia, we can just follow the pedigree, the outward phenotype, and then we can follow the molecular phenotype, and we can do all kinds of predictive things. You need to do that using a DNA blot, because you have to detect it with a specific probe for that gene, or by using the polymerase chain reactions. Here's a situation in which you have a polymorphism between a disease gene, cystic fibrosis, and a normal allele, non-cystic fibrosis. Here's where the polymorphic site is right here. In order to use PCR, you need to amplify this region. You need to amplify it using primers that are common to both alleles, not different. You amplify up the DNA, and then you digest it with the restriction enzyme and put it on a gel. Look at the different sizes. Here's the cystic fibrosis individual. Here's the individual that has the normal allele. Here's the heterozygote. Because you can do this with PCR, it's very powerful because you can detect prenatally or pre-implantation.